Okay, our next presenter is Tawni Riepe. She's a second-year master's student in fish, wildlife, and conservation biology. And this is someone who really had found their life's calling as a child, like some of our others. Her playgrounds were creeks, ponds, swamps, and reservoirs, which led to her current interest in fisheries science and disease ecology. And I'll also point out that she works with a lot of fish. She also is a sport fisher person for trout, muskie, and bass, okay, which I think is absolutely amazing. And the title of her presentation is A So-FISH-ticated Disease.
Imagine yourself fishing on a boat in the middle of summer underneath the beautiful Colorado sky. As you make one of your many casts, you finally get a tug at the end of the line. You net it and get it into your boat, but, gross. All you're staring at is this sick fish riddled with disease. Diseases in fish are often first managed on a hatchery unit. Hatcheries are these facilities that raise many of our sport fish we like to catch, such as rainbow trout, and many of our rarer threatened species, such as the Colorado native greenback cutthroat trout. Once they're raised, they're then released into rivers and reservoirs throughout the state. But prior to release, the fish need to go through a series of tests to ensure that they are healthy and free from pathogens. Pathogens are these organisms, such as a bacteria, that actually cause the disease associated with it. One such pathogen is called Renibacterium salmoninarum, which I'm going to call RSAL for short, and it is a bacteria that causes bacterial kidney disease in many trout and salmon species, contributing to high rates of death and is often considered very sophisticated. RSAL is considered sophisticated because it has this ability of hiding itself inside of a white blood cell. White blood cells are found throughout the body and often respond to any sort of attack on the immune system. So if you can imagine a white blood cell like a Pac-Man, it goes around the body gobbling up anything that's not supposed to be there and destroying it, such as a bacteria. So when a white blood cell comes in contact with an RSAL bacteria, RSAL actually tricks it so that it gets gobbled up but not destroyed. Instead, it stays inside and replicates itself, making it hard for the immune system to detect it and eventually causes disease in that fish, maybe some years down the road. So to ensure that fish remain healthy on the hatchery unit, I'm most interested in figuring out how we should actually be testing for this RSAL bacteria within the fish. Now, given the name of the disease associated with it, bacterial kidney disease, protocols do suggest that we test the kidney tissue, but since we know it is so efficient at hiding in those white blood cells, we're wondering if we can find it in other parts of the body as well. So we went and collected 800 greenback cutthroat trout, tested 6 different tissues from each one, including the liver, spleen, and kidney, blood samples, ovarian fluid, and mucus. And to our surprise, we actually saw a higher prevalence of that bacteria in the liver tissue, not the kidney. Understanding how to best detect for this bacteria in the fish will help aid in future disease management protocols so that when those hatchery fish are released into the wild, we can be certain that they are healthy and free from RSAL infections. So then when fishermen go out, they can enjoy catching a beautiful trophy trout, not one that's going to make them go, ugh, gross. Thank you.